The reef does not erupt in motion. There is no burst of sand, no desperate jet of ink, no flicker of fleeing arms. One moment, the octopus rests on open rock, its body clearly defined against pale limestone and scattered coral fragments. And the next moment, it is no longer there. It does not retreat. It does not hide behind an object. It simply dissolves into the texture of the reef itself, as if the boundary between animal and environment has collapsed. What remains in the frame is only stone, shadow, and the faint illusion of uneven color. The transformation is not an act of disappearance in space, but an act of disappearance in perception. The animal has not moved. Instead, the world around it has been absorbed into its skin. Beneath that skin lies a biological system so intricate that it functions less like pigment and more like computation. The octopus does not possess scales or fur to alter its outline. It carries an adaptive surface constructed from layers of specialized cells that operate in coordination. The outermost layer contains chromatophores, tiny elastic sacs filled with pigment and surrounded by radial muscles. When these muscles contract, the pigment sac expands, spreading color across the skin in fractions of a second. Each chromatophore is individually controlled by direct neural input, allowing for fine-scale patterning that can replicate the mottled surfaces of coral rubble or the smooth gradations of sand. Below them sit iridophores, which reflect light through microscopic platelets that scatter and refract specific wavelengths, producing iridescent blues and greens that cannot be achieved through pigment alone. Deeper still are leucophores, cells that diffuse ambient light, effectively sampling the environment's brightness and feeding it back as a neutral background tone. These layers do not operate independently. They form a living screen, stacked and interlocked, capable of generating both color and reflectance simultaneously. What makes this system extraordinary is not only its complexity, but its speed. Within moments of landing on a new substrate, the octopus matches the brightness, contrast, and spatial frequency of the surrounding terrain. It reads the reef as a mosaic of shapes and tonal gradients, then projects a corresponding image across its own body. The precision suggests vision-driven analysis, yet the mechanics extend beyond simple sight. The octopus eye gathers information about contrast and polarization, feeding it to a brain that is already primed to decode texture. But here lies the deeper revelation. The octopus does not centralize all control within its head. A vast portion of its neurons reside in its arms, which act semi-autonomously, capable of processing tactile information and adjusting local skin patterns without waiting for top-down instruction. The skin, in effect, participates in perception. It is not a passive canvas, it is an active responder. As the animal settles into the reef, papillae begin to rise along its mantle and arms. These muscular hydrostats alter the physical topography of the skin, creating three-dimensional structures that mimic the bumps and ridges of coralline rock. Camouflage here is not limited to color matching. It is sculptural. The smooth outline of the octopus becomes fractured and uneven, breaking up its silhouette into irregular fragments that merge with surrounding debris. Predators scanning for recognizable shapes encounter only visual noise. Prey, searching for shelter, drift unaware across what they perceive as inert substrate. This capacity for texture mimicry demonstrates that concealment in the marine environment is not solely a matter of blending hues, but of dissolving form. In shallow tropical reefs, light shifts constantly as waves ripple above. Patterns flicker across the seabed in moving bands of brightness known as caustics. The octopus does not resist these changes. It synchronizes with them. Its skin pulses with alternating light and dark patches, echoing the dynamic shimmer of water overhead. This responsiveness indicates that camouflage must operate within an ecological context that includes motion and depth. A static pattern would betray the animal the moment the environment changes. Instead, the octopus maintains a feedback loop with its surroundings, adjusting pattern amplitude and contrast as light conditions fluctuate. The result is a living surface that remains consistent with the reef's shifting optics. Predator-prey dynamics further refine this strategy. Reef fish rely heavily on edge detection to identify threats. Their visual systems are tuned to detect abrupt transitions between object and background. By fragmenting its outline into discontinuous shapes, the octopus undermines this detection mechanism. Its body becomes a field of broken patches rather than a coherent figure. Meanwhile, when stalking prey, it employs a different tactic known as disruptive coloration, creating bold, high-contrast patterns that mimic the natural shadows of coral branches. As it creeps forward with careful, undulating movements, its skin absorbs the micro-textures of the substrate. The arms extend gradually, their tips exploring crevices for hidden crustaceans. At the final moment, the octopus abandons invisibility and erupts into motion, enveloping its prey in a coordinated surge of muscular precision. The sophistication of this system reflects evolutionary pressures unique to soft-bodied predators. Lacking the protective armor of many reef organisms, the octopus must rely on concealment rather than confrontation. Its vulnerability has driven the development of a camouflage network that integrates vision, neural processing, muscular control, and optical physics into a unified survival mechanism. Unlike animals that rely on seasonal color change or limited pattern shifts, the octopus can alter its appearance dozens of times within minutes, responding to every microhabitat it encounters. This adaptability grants access to a wide range of ecological niches, from sandy flats to complex coral mazes. Recent studies suggest that octopus skin may even possess light-sensitive proteins similar to those found in the retina. While the extent of this capability remains under investigation, evidence indicates that the skin itself can detect changes in brightness and adjust chromatophore expansion accordingly. If confirmed, this would imply a distributed sensory network embedded across the body surface. Camouflage would then be governed not solely by visual input from the eyes, but by localized photoreception. The boundary between seeing and being seen would blur further, reinforcing the idea that the octopus operates as a decentralized intelligence. Thermoregulation and metabolic efficiency also intersect with this camouflage system. By adjusting pigmentation and reflectance, the octopus can influence heat absorption in shallow waters where temperature fluctuates with sunlight exposure. Darker patterns may enhance heat uptake during cooler periods, while lighter tones reflect excess radiation. Though camouflage remains the primary function, these secondary effects reveal the interconnected nature of physiological adaptation. No trait evolves in isolation. Coloration participates in multiple survival equations simultaneously. Migration patterns in octopus species are generally limited compared to pelagic fish, yet their habitat flexibility is remarkable. A single individual may traverse rocky outcrops, sandy expanses, and seagrass beds within a small territory. Each transition demands immediate recalibration of skin pattern and texture. The energy cost of maintaining such responsiveness is significant, supported by a high metabolic rate relative to body mass. Oxygen consumption increases during active camouflage shifts, reflecting the muscular effort required to expand chromatophores and erect papillae. The octopus balances this expenditure by alternating periods of intense activity with stillness, during which its camouflage stabilizes into a low-energy state. Within the reef community, this ability reshapes ecological relationships. Cleaner fish, which often approach larger animals to remove parasites, hesitate when confronted with an octopus that appears indistinguishable from rock. Small crustaceans that normally rely on color vision for predator detection fail to register the threat until it is too late. Even human divers, trained to identify marine life, often swim past an octopus without recognition. The animal's success lies not in hiding behind objects, but in becoming an object. The moment of vanishing that opened this scene is not a theatrical display. It is a biological necessity forged through millions of years of evolutionary experimentation. The octopus survives not because it is invisible in the absolute sense, but because it manipulates the sensory systems of others. It studies the reef through light, texture, and motion, then feeds that information back across its own body in a seamless exchange. The result is a predator that does not occupy a fixed identity within the landscape. It exists as a transient echo of its surroundings, constantly rewritten by environmental input. When the octopus eventually moves again, lifting from the rock with a slow undulation of arms, its colors shift once more, abandoning the previous substrate and adopting a new pattern suited to open water. The reef reclaims its appearance as stone and coral, and the illusion ends only because the animal chooses to reveal itself. Camouflage here is not a static mask, but a conversation between organism and ecosystem. The skin thinks, the arms decide, and the reef becomes indistinguishable from the predator that inhabits it. A ripple of light travels across the seabed, but it does not come from the sun above. It originates from a hovering body suspended just centimeters above the sand, where a cuttlefish holds position with delicate undulations of its lateral fins. Across its skin, bands of brightness sweep from head to tail in rhythmic pulses, like luminous tides contained within living tissue. Small crabs freeze mid-step, their compound eyes detecting movement, yet failing to interpret its meaning. A juvenile fish hesitates, caught between curiosity and caution, as the cuttlefish's patterns intensify, then soften, then flare again in precise waves. The prey has not yet recognized a predator. It is processing a sensory anomaly. Within seconds, the cuttlefish surges forward, tentacles launching with ballistic precision, and the illusion collapses into a successful strike. What began as a display of shifting color was not camouflage in the conventional sense. It was a neurological intervention. The cuttlefish inhabits coastal reefs, seagrass meadows, and sandy lagoons, where visibility fluctuates with water clarity and light penetration. Unlike species that rely solely on blending into static backgrounds, cuttlefish employ a strategy best described as dynamic camouflage, a process that merges concealment with active visual signaling. Their skin contains chromatophores, similar to those of octopuses, but the organization and neural control allow for extraordinary spatial and temporal patterning. Each chromatophore is a pigment sac surrounded by radial muscles that expand or contract under direct neural control. Beneath these lie iridophores and leucophores that reflect and scatter light, contributing to structural coloration and brightness matching. What distinguishes cuttlefish is the speed and coordination with which these elements generate moving patterns that travel across the body like waves of electricity. In hunting scenarios, these patterns often take the form of rhythmic pulses known as passing clouds. Dark and light bands sweep laterally across the mantle, creating a flickering effect that appears almost hypnotic. Research suggests that such displays may exploit motion-sensitive pathways in prey species, overwhelming their visual processing systems. Small fish and crustaceans rely on rapid assessment of contrast and movement to detect threats. When confronted with oscillating patterns that mimic environmental fluctuations, their neural circuits may struggle to categorize the stimulus as predator. The cuttlefish effectively introduces noise into the perceptual field, delaying recognition long enough to close the distance. This strategy reveals a deeper principle of predator-prey interaction. Survival often hinges not only on physical capability, but on control of information flow within sensory systems. As the cuttlefish drifts closer to a cluster of shrimp, its body begins to flatten slightly, reducing vertical relief and casting minimal shadow. The dynamic patterns gradually transition into a modeled arrangement that mirrors the grain of the substrate. Unlike static camouflage, which locks an animal into a single appearance, dynamic camouflage allows continuous adjustment to microhabitats encountered during movement. The cuttlefish does not commit to a single disguise. It updates its projection in real time, responding to variations in sand texture, seagrass density, and light intensity. This flexibility is crucial in shallow waters, where shifting currents alter sediment distribution and refract sunlight into constantly changing caustic patterns. The cuttlefish's large, W-shaped pupils play a significant role in this adaptive process. These specialized eyes provide wide-angle vision and enhanced contrast detection, enabling the animal to analyze spatial frequencies in the environment. By assessing the scale of patterns around it, the cuttlefish can match not only color, but also the size and orientation of surrounding textures. This is particularly important in seagrass meadows, where vertical blades create linear shadows that differ markedly from the granular appearance of sandy flats. Through rapid neural processing, the cuttlefish adjusts chromatophore expansion to align with these environmental cues, minimizing edge detection by predators. Predators of cuttlefish include larger fish, marine mammals, and occasionally seabirds. When the animals venture into shallows, the cuttlefish's response to threat often begins with a sudden shift to high-contrast disruptive patterns that break up its outline. Rather than blending seamlessly, it fragments its body into bold patches of dark and light, confusing the predator's ability to track a coherent shape. If the threat persists, the cuttlefish may employ a diamatic display, rapidly expanding dark eye spots and increasing body size through mantle inflation. This abrupt visual escalation can startle predators long enough for escape via jet propulsion. The sequence demonstrates a layered defense strategy, moving from subtle deception to overt warning, depending on the immediacy of danger. Beyond camouflage and intimidation, cuttlefish utilize color change in complex social interactions. During mating season, males often display vivid, high-contrast patterns to attract females, while simultaneously concealing their presence from rival males. In a behavior known as split signaling, a male may present courtship patterns on the side facing a female and subdued, female-like coloration on the side visible to competitors. This dual presentation exploits the directional vision of other cuttlefish, effectively allowing the male to broadcast different messages simultaneously. The sophistication of this behavior underscores the neural capacity required to manage independent patterning across the body's surface. Thermoregulation intersects subtly with these visual adaptations. In shallow coastal habitats, temperature can fluctuate significantly between day and night. By altering pigmentation and reflectance, cuttlefish may influence heat absorption, with darker patterns potentially increasing solar gain during cooler periods. Although this function is secondary to camouflage and communication, it illustrates the multifunctional nature of color change. Evolution tends to favor traits that serve multiple ecological roles, maximizing efficiency within energetic constraints. Cuttlefish are relatively short-lived, with lifespans often limited to 1 or 2 years, depending on species. This brevity places intense selective pressure on rapid growth, efficient feeding, and successful reproduction. Dynamic camouflage contributes directly to feeding efficiency by enhancing hunting success and reducing predation risk during vulnerable juvenile stages. Young cuttlefish, only a few centimeters in length, already possess the neural architecture necessary for complex patterning. Observations of juveniles reveal early deployment of disruptive patterns when startled, indicating that these capabilities are innate rather than learned through experience. In the broader ecological context, cuttlefish occupy a mid-level predatory role, feeding on crustaceans, small fish, and other invertebrates, while serving as prey for larger animals. Their capacity to manipulate perception influences energy flow within the ecosystem. By increasing hunting efficiency, they regulate populations of small invertebrates, indirectly affecting algal growth and sediment turnover. At the same time, their ability to evade predators ensures sufficient survival to sustain higher trophic levels. Camouflage here is not merely a personal defense, it shapes community dynamics. Light conditions further refine the cuttlefish's visual strategies. In deeper or murkier waters, where red wavelengths diminish and blue dominates, the animal adjusts reflectance through iridophores to maintain appropriate brightness contrast. Some species can even reflect polarized light, a property that may be visible to other cephalopods, but less detectable to many fish. This suggests a private communication channel layered atop public camouflage, allowing subtle signaling without compromising concealment. As dusk approaches, the reef undergoes a transition. Diurnal fish retreat into crevices, and nocturnal predators emerge. The cuttlefish responds by reducing high-frequency patterning and adopting darker, more uniform coloration suited to low-light conditions. In these moments, dynamic camouflage shifts, emphasis from visual deception to shadow management. The animal positions itself relative to moonlight and bioluminescent cues, minimizing silhouette against the water column. Although its color displays are less dramatic at night, the underlying neural choreography continues, constantly sampling and adjusting. In the end, the cuttlefish's mastery lies not solely in disappearing into its surroundings, but in orchestrating the visual field of others. It modulates brightness, contrast, motion, and pattern to either conceal its presence or command attention, depending on ecological need. The flashing waves that first appeared as aesthetic spectacle are, in fact, calculated manipulations of sensory perception. Prey are delayed in recognizing threat, predators are momentarily confused, and rivals are deceived through selective signaling. Each pattern change reflects an integration of environmental data, neural processing, and muscular precision. As the cuttlefish glides away from the hunting ground, its skin transitions to a subdued modeling that mirrors the fading light. The pulses subside, leaving only subtle shifts that echo the grain of the sand. What remains is a creature that has evolved not merely to inhabit its environment, but to rewrite how that environment is seen by others. In coastal ecosystems, defined by fluctuating light and complex interactions. The cuttlefish stands as a reminder that survival often depends on mastering perception itself. A juvenile dam sailfish hovers just above the reef, its movements small and rhythmic as it picks at drifting plankton. The current is gentle, pushing fragments of algae and suspended particles across a patchwork of coral rubble. Nothing appears unusual in the frame. The reef is crowded with shapes, encrusting sponges, branching corals, patches of filamentous growth swaying in slow arcs. The fish shifts slightly to the left, then forward, unaware that the object it has been passing for several minutes, a knotted, irregular mass coated in mottled reds and browns, is not coral at all. There is no warning flash, no chase sequence. The water seems to fold inward for a fraction of a second, and the damselfish vanishes. The coral remains where it was. Only after careful inspection does the outline resolve into the thick-lipped mouth of a frogfish, now distended and settling back into its original contour. The predator had been visible the entire time, but it did not register as an animal. It existed as background. Frogfish belong to the family Antennareidae, a group of anglerfish adapted to shallow tropical and subtropical reefs. Unlike their deep-sea relatives, which rely on bioluminescent lures in darkness, frogfish operate in well-lit environments, where camouflage must withstand scrutiny from visually acute prey and predators alike. Their survival strategy rests not on speed or endurance, but on absolute stillness and morphological deception. The frogfish does not resemble a fish in the conventional sense. Its body is globular, often compressed laterally, and supported by modified pectoral fins that function like limbs. These fins allow it to walk slowly across the substrate, repositioning without attracting attention. The skin is thick and covered with dermal spinules and filamentous appendages that break up the outline into irregular textures, closely mimicking sponges, tunicates, or encrusting algae. The illusion begins with color. Chromatophores embedded within the skin allow frogfish to shift pigmentation over days or weeks, gradually matching the dominant tones of their chosen habitat. This process is slower than the rapid color change of cephalopods, but it reflects a longer-term commitment to a specific microenvironment. Once settled, a frogfish may remain in the same area for extended periods, adjusting its coloration to align with seasonal changes in coral growth or algal cover. The resulting camouflage is not dynamic in the moment-to-moment sense, but it is precise and durable. From a distance, the animal's surface appears as a continuation of the reef matrix, complete with subtle shadows and raised textures that mimic living coral polyps. Texture is equally critical. Many species develop fleshy appendages that resemble sponge oscula or branching hydroids. These projections are not random. Their distribution corresponds to the structural complexity of surrounding organisms. In reefs dominated by filamentous algae, frogfish often exhibit elongated skin flap that sway gently with water movement, reinforcing the illusion of plant life. In areas rich in encrusting sponges, their bodies become knobbier and more irregular. The interplay between morphological plasticity and habitat selection reveals a refined evolutionary dialogue between predator and environment. Individuals that most closely matched their surroundings were more likely to survive and reproduce, gradually shaping a lineage that blurs the boundary between animal and substrate. Stillness is the final component of this deception. Many reef predators rely on detecting motion rather than color contrast alone. By remaining motionless for extended periods, Frogfish minimize the cues that would reveal them as living organisms. Their respiration is subtle, achieved through small gill openings located behind the pectoral fins, reducing visible movement at the mouth. When prey approach, the frogfish does not track them with overt head turns. Instead, its eyes, capable of independent movement, adjust slowly within their sockets, maintaining surveillance without altering the body's silhouette. The ecological principle here is straightforward. In complex visual environments, the absence of movement often renders an object effectively invisible. The feeding mechanism that follows this prolonged invisibility is among the fastest in the animal kingdom. Frogfish possess an extensible mouth capable of expanding to several times its resting volume. When prey cross an invisible threshold, specialized muscles depress the lower jaw, while the buccal cavity expands rapidly, generating a sudden drop in pressure. Water and prey are drawn inward in a suction event that unfolds in milliseconds. The speed of this strike leaves little opportunity for escape. The prey is engulfed before its nervous system can initiate a response. High-speed imaging reveals that the entire sequence, from initiation to closure, occurs in less than a blink. This extreme specialization compensates for the frogfish's lack of pursuit capability. It does not chase. It waits for the environment to deliver food within range. Central to this ambush strategy is the elytium, a modified dorsal spine that functions as a lure. At its tip lies the esca, a fleshy appendage that often resembles a small worm or shrimp. The frogfish manipulates this lure with delicate movements, oscillating it just above the substrate to simulate living prey. Small fish and crustaceans, drawn by what appears to be an easy meal, approach cautiously. The irony is biological rather than humorous. The predator attracts prey by imitating the prey's own food sources. This layered deception underscores the complexity of trophic interactions within reefs. The frogfish inserts itself into existing feeding circuits, exploiting behavioral instincts honed by evolution. Predators of frogfish include larger fish and occasionally octopuses. Against such threats, camouflage again becomes the primary defense. Some species are capable of gulping water to inflate their bodies, increasing apparent size and making ingestion more difficult. However, this is a secondary measure. Avoidance through invisibility remains the dominant strategy. The frogfish's thick skin and lack of scales also provide some protection against abrasion and minor attacks, reflecting adaptation to a sedentary lifestyle among sharp coral fragments. Thermoregulation in frogfish is largely governed by environmental temperature, as they are ectothermic. Their metabolic rate fluctuates with water temperature, influencing feeding frequency and growth. In warmer waters, digestion accelerates, enabling more frequent strikes. However, increased metabolism also raises oxygen demand, which must be met through efficient gill ventilation, despite minimal body movement. This constraint reinforces the importance of ambush efficiency. Each feeding opportunity must yield maximum energetic return to sustain the frogfish's relatively slow but steady growth. Reproductive behavior introduces a temporary departure from stillness. During spawning events, male and female frogfish ascend toward the surface in synchronized movements, releasing gametes into the water column. The eggs are encased in buoyant mucus ribbons that drift with currents, dispersing larvae over broader areas. This pelagic phase contrasts sharply with the adult's benthic existence, allowing colonization of new reef systems. Once settled, juveniles begin the process of habitat matching, selecting substrates that align with their developing morphology. Over time, individual frogfish may shift locations if environmental conditions change, but such movements are deliberate and infrequent. Within the broader reef ecosystem, frogfish serve as regulators of small fish populations. Their presence influences prey distribution, encouraging schooling behavior and habitat shifts that ripple through trophic levels. By removing individuals from local populations, frogfish indirectly affect algal grazing patterns and coral competition. Though they occupy a single node within a vast ecological network, their impact is measurable. The invisibility that defines their hunting strategy does not equate to ecological insignificance. As the reef transitions from midday brightness to the softer hues of late afternoon, the frogfish remains in place. Light angles change, shadows lengthen, and plankton density fluctuates with tidal flow. The predator's coloration, already aligned with its chosen coral head, continues to blend seamlessly. A passing rasa inspects the substrate, unaware that it hovers within striking distance. The frogfish's lure twitches once, twice, imitating the erratic movement of a small invertebrate. The rasa edges closer, drawn by instinct. In the blink of an eye, the reef appears unchanged. Yet another fish has disappeared into a mouth that never seemed to exist. The frogfish does not rely on spectacle or rapid pursuit. Its success is rooted in absence of motion, absence of recognizable shape, absence of visual cues that would signal threat. By becoming indistinguishable from coral or sponge, it effectively removes itself from the perceptual map of surrounding animals. The reef is not merely its habitat, it is its identity. In ecosystems defined by intense competition and predation, the frogfish demonstrates that power can reside in invisibility, and that the most effective predator is sometimes the one that never appears to be present at all. The seabed appears empty, a mosaic of pale sand interrupted by scattered rubble and low coral outcrops. Sunlight filters through clear tropical water, casting rippling patterns that dance across the substrate. A school of goatfish drifts overhead, sifting through sediment in search of invertebrates. A curious rasi darts between stones, probing crevices with quick, precise movements. Nothing in this quiet scene suggests immediate danger, and yet, resting only a few centimeters away from the rasi's path, lies one of the most venomous vertebrates in the ocean. It is fully visible, illuminated by the same sunlight that touches every grain of sand, but the eye refuses to acknowledge it. The stonefish does not hide behind rock. It has become rock. Only when a fin brushes too close or a careless step presses downward does the illusion shatter. And by then, the encounter has already shifted from observation to consequence. The stonefish inhabits shallow coastal reefs, lagoons, and sandy flats throughout the Indo-Pacific region, where water temperatures remain warm year-round, and coral growth creates a complex visual environment. Its body is squat and irregular, covered in warty protuberances and ridges that replicate the fractured geometry of reef substrate. Unlike streamlined fish, built for speed, the stonefish appears almost unfinished, as if shaped from the same calcified fragments scattered around it. Pigmentation ranges from mottled browns and grays to muted reds and yellows, often overlaid with algae that colonize its skin. This encrusting growth is not incidental. It enhances the disguise, transforming the fish into a living extension of the reef. At first glance, camouflage in the stonefish serves an obvious predatory function. By remaining motionless and indistinguishable from surrounding rock, it allows small fish and crustaceans to approach within striking distance. Its feeding mechanism relies on suction, similar to that of frogfish, but with a broader gape relative to body size. When prey drift too close, the stonefish depresses its lower jaw and expands its buccal cavity, generating a rapid influx of water that pulls the target inward. The strike unfolds in fractions of a second, leaving little time for escape. Yet this is only part of the story. Camouflage here operates not merely to secure meals, but to reduce detection by predators and minimize energy expenditure in an environment where constant vigilance would be metabolically costly. The stonefish's venom apparatus introduces a defensive dimension that distinguishes it from many other ambush predators. Along its dorsal surface, 13 stout spines stand erect, each connected to venom glands housed at the base. When pressure is applied, such as from a predator's bite or an unwary foot, the spines penetrate flesh and deliver venom through grooves along their length. The venom is a complex mixture of proteins and peptides capable of causing intense pain, tissue damage, and in severe cases, systemic effects. For the stonefish, this defense is passive. It does not pursue confrontation. It waits for contact. The irony is stark. The fish most difficult to see is also among the most dangerous to touch. This dual strategy of invisibility and venom reflects evolutionary trade-offs. A sedentary lifestyle reduces caloric demand but increases vulnerability to predators that rely on olfactory cues or accidental discovery. By coupling camouflage with a potent deterrent, the stonefish mitigates the risks associated with immobility. Predators that attempt to consume it often learn quickly to avoid similar shapes in the future, reinforcing the effectiveness of its disguise through experiential memory. In this way, the stonefish influences predator behavior across the reef, contributing to a subtle shaping of feeding patterns. Environmental context enhances the camouflage. Coral reefs are dynamic systems where light refracts through surface waves, creating shifting mosaics of brightness and shadow. The stonefish's irregular surface breaks up reflected light, preventing consistent highlights that would betray a smooth, biological form. Fine sediments settle into creases along its body, further aligning it with the substrate. Even experienced divers, trained to identify marine life, often overlook stonefish resting in plain view. Detection typically occurs only when movement betrays the outline, or when careful inspection reveals the slight rise and fall of gill openings. Respiration itself is minimized to preserve the illusion. Instead of continuous mouth movement, the stonefish relies on opercular openings behind the head to draw water across its gills. These subtle currents are less conspicuous than open-mouth ventilation. The eyes, positioned high on the head, allow the fish to monitor its surroundings without altering body posture. Vision plays a critical role in timing strikes, yet the stonefish does not track prey with overt movements. It waits for prey to cross a narrow arc in front of its mouth, then releases stored muscular energy in a sudden expansion. Thermoregulation in stonefish is largely dictated by ambient water temperature, as with most marine fishes. However, their preference for shallow habitats exposes them to fluctuations associated with tidal cycles and solar heating. During periods of extreme heat, stonefish may retreat to slightly deeper zones or seek shaded areas beneath coral ledges. This microhabitat selection reflects a balance between optimal hunting grounds and physiological tolerance. Warmer water accelerates metabolic processes, increasing oxygen demand. Given their reliance on stillness, stonefish must ensure that oxygen availability remains sufficient without compromising camouflage through excessive movement. Reproductive behavior introduces a contrasting dynamic to their otherwise static existence. During spawning events, stonefish release buoyant egg masses into the water column, where fertilization occurs externally. The eggs drift with currents, dispersing larvae across broader reef systems. This pelagic phase reduces intra-specific competition and facilitates colonization of new habitats. Juvenile stonefish settle onto substrates that match their developing coloration, gradually refining their camouflage as they grow. Over time, individuals may shift habitats slightly if sediment composition changes, demonstrating a degree of plasticity within their sedentary framework. Predator-prey interactions extend beyond simple ambush. Stonefish occupy a niche that influences the distribution of small reef fish. Areas with high stonefish density may exhibit altered schooling patterns as prey species learn to avoid specific substrate types or regions of the reef. This behavioral adaptation can cascade into changes in grazing pressure on algae and competition among invertebrates. Thus, the stonefish's invisibility contributes indirectly to broader ecological dynamics, shaping energy flow and habitat use. Human interactions underscore the effectiveness of the stonefish's camouflage. Accidental envenomations occur when swimmers or divers step on fish concealed in shallow water. The intense pain associated with stings has earned the stonefish a formidable reputation. Yet the fish does not actively seek contact. Its defense is triggered only by pressure. In many documented cases, victims report that they did not perceive the animal until after contact occurred. This highlights the distinction between visibility and recognition. The stonefish can be present within the field of vision, yet remain cognitively absent until the consequence reveals its existence. The stonefish's evolutionary lineage traces back to scorpion fish, sharing morphological traits such as venomous spines and cryptic coloration. Over time, selective pressures in shallow reef environments favored individuals that minimized motion and maximized resemblance to inert substrate. The result is a fish that has sacrificed speed and agility for deception and defense. Its musculature supports rapid jaw expansion, but limited sustained swimming. Energy allocation prioritizes venom production and tissue maintenance over locomotor endurance. As twilight approaches, the reef's color palette shifts toward muted blues and grays. The stonefish remains in place, its outline merging even more seamlessly with fading light. Nocturnal predators begin to patrol, and plankton rise in the water column. The stonefish's hunting window extends into darkness, where contrast diminishes and prey rely more heavily on lateral line detection. Yet even here, stillness remains effective. Without movement to trigger mechanosensory alarms, the predator remains undetected. The most profound aspect of the stonefish's strategy lies in its refusal to participate visibly in the ecosystem's constant motion. While other predators chase, stalk, or flash warning colors, the stonefish opts for erasure. It occupies space without announcing presence, relying on a fusion of morphology, pigmentation, and venom to secure survival. Its invisibility serves both to capture prey and to deter predators, embodying a dual-purpose adaptation refined through evolutionary time. In the calm of the lagoon, where currents whisper across sand and coral, casts intricate shadows, the stonefish persists as a quiet hazard. It does not advertise danger, it conceals it. Only when the substrate itself seems to bite does the reef reveal the predator embedded within. By then, the lesson has been delivered through pain or loss, and the stonefish resumes its patient vigil, once again indistinguishable from the world around it. The current moves first, not the animal. A slow pulse of water slides across the reef edge, bending strands of kelp and ribbons of brown algae into synchronized arcs. Sunlight filters down in narrow shafts, fractured by surface ripples, casting wavering shadows over a forest of swaying vegetation. Within that living tapestry, a fragment of seaweed detaches and drifts a few centimeters higher than the rest. It tilts slightly, rotates, then settles back into the rhythm of the surge. Only when the eye lingers does the fragment resolve into something structured. An elongated snout, a dark, deliberate eye, a torso suspended vertically. The leafy sea dragon has not entered the frame. It has always been here. It is not hiding behind the kelp. It is participating in it. The leafy sea dragon inhabits temperate coastal waters along southern Australia, particularly in regions dominated by kelp forests and seagrass meadows. These environments are defined by vertical complexity and perpetual motion. Kelp fronds rise from rocky substrates, their buoyant blades swaying with tides and swell, creating a dynamic visual field where shape and orientation shift continuously. In such habitats, traditional camouflage based on static background matching would be insufficient. The background itself is in motion. The leafy sea dragon's evolutionary response has been to align not only its coloration with this environment, but its entire body architecture and behavior with the fluid geometry of drifting algae. Its most striking features are the elaborate dermal appendages that extend from its head, torso, and tail. These leaf-like projections resemble torn strips of seaweed, branching and tapering in irregular patterns. Importantly, these structures contain no muscular control for propulsion. They are not fins in the conventional sense. Locomotion is achieved through small, nearly transparent pectoral, toral, and dorsal fins that beat rapidly and subtly, generating propulsion without disturbing the overall silhouette. The appendages exist purely for deception. From an energetic standpoint, they represent a significant investment in tissue that does not contribute directly to swimming efficiency. Yet in the ecological context of kelp forests, where predation pressure is high and visibility fluctuates with turbidity, the advantage of blending seamlessly into moving vegetation outweighs the cost. The body of the leafy sea dragon is laterally compressed and held in a near vertical orientation, mimicking the posture of upright seaweed blades. Pigmentation further enhances this illusion. Yellows, olive greens, and muted browns dominate its surface, often accented with subtle striping that mirrors the natural banding of algae. Unlike cephalopods, which alter coloration rapidly, the sea dragon's hues are relatively stable, shaped by longer-term adaptation rather than instantaneous change. This stability reflects its specialized niche. It does not roam widely across varied substrates, but remains within habitats where kelp and seagrass provide consistent visual structure. Predator avoidance is the primary driver of this extraordinary morphology. In temperate coastal ecosystems, larger fish and marine mammals patrol kelp forests in search of prey. Visual detection remains a dominant hunting strategy, even in waters where light penetration is reduced by plankton blooms or suspended sediment. The leafy sea dragon disrupts the recognition process by fragmenting its outline into irregular, plant-like segments. Predators scanning for coherent fish shapes encounter instead a scatter of leaf fragments aligned with the surrounding vegetation. The animal does not conceal itself beneath cover. It erases the concept of fish from the visual equation. Behavior reinforces morphology. Rather than swimming in straight lines or making abrupt directional changes, the leafy sea dragon allows currents to dictate much of its movement. It drifts, rotates gently, and adjusts position with minimal fin beats. This passive locomotion is not weakness, but strategy. In kelp forests, the most conspicuous element is often the organism that resists the sway of water. By moving in harmony with the current, the sea dragon maintains consistency with background motion. The illusion depends on synchronization between body and environment, a subtle choreography that has been refined through natural selection. Feeding introduces a delicate tension between deception and action. The leafy sea dragon preys primarily on small crustaceans, such as mysid, shrimp, and planktonic invertebrates. Its elongated snout houses a tubular mouth designed for suction feeding. When prey drift within range, the sea dragon generates a rapid intake of water, drawing the organism inward without lunging or twisting its body. The strike is subtle and localized, preserving the overall impression of passive vegetation. Even in feeding, the animal avoids gestures that would compromise its disguise. Reproduction reveals another layer of biological specialization. Unlike many fish species, male leafy sea dragons assume the responsibility of carrying fertilized eggs. After courtship, the female deposits bright pink eggs onto a specialized brood patch located beneath the male's tail. The eggs adhere to this vascularized area, where they receive oxygen and protection for several weeks. Throughout this period, the male continues to inhabit kelp forests, relying on camouflage to shield himself and his developing offspring. The brood patch itself becomes integrated into the overall illusion, its coloration blending with the surrounding appendages. This reproductive strategy reflects a trade-off between mobility and parental investment. By remaining concealed within vegetation, the male reduces predation risk during a vulnerable life stage. Thermoregulation in temperate waters presents different challenges than in tropical reefs. Seasonal fluctuations in temperature can influence metabolic rate and prey availability. Leafy sea dragons exhibit limited migratory behavior, often remaining within relatively small home ranges, where kelp density provides consistent shelter. During colder months, metabolic processes slow, reducing feeding frequency and growth rate. Warmer periods may increase activity levels, but the animal rarely ventures far from vegetated habitats. Its survival is tightly linked to the health of kelp ecosystems, which themselves are sensitive to climate variability and human impact. The broader ecological role of the leafy sea dragon extends beyond its individual survival. By preying on small crustaceans, it participates in regulating plankton populations that influence nutrient cycling within coastal waters. Its presence also serves as an indicator of kelp forest health. Declines in sea dragon populations often correlate with habitat degradation, whether from rising sea temperatures, pollution, or physical disturbance. Thus, the animal's camouflage not only ensures personal survival, but also ties it intimately to ecosystem stability. Sensory adaptations complement visual deception. The leafy sea dragon relies on well-developed eyesight to locate prey among drifting particles. Its independently moving eyes allow it to scan the environment without altering body position. Lateral line systems detect subtle water movements, providing information about nearby organisms. These sensory inputs enable precise feeding strikes while maintaining minimal overt motion. The integration of perception and stillness underscores the complexity of its ecological strategy. Human observers often describe encounters with leafy sea dragons as surreal, a moment of realization when a patch of algae acquires depth and intent. This perceptual delay highlights the effectiveness of its disguise. Even in controlled settings where divers know to expect its presence, the animal can remain unnoticed until proximity or movement reveals it. The deception operates not through invisibility in a physical sense, but through cognitive misdirection. The brain categorizes the appendaged form as plant life until contradictory evidence forces reinterpretation. As dusk settles over the kelp forest, light angles steepen and shadows lengthen. The sea dragon drifts among fronds that darken from olive to near black. Plankton rise in the water column, and the animal continues its quiet feeding. Its appendages cast intricate silhouettes that merge with the tangled vegetation. In this dimming world, the boundary between organism and environment dissolves further. The leafy sea dragon does not retreat to a crevice or burrow into sand. It remains suspended in open water, confident in the architecture of its disguise. The evolutionary pathway that produced this living fragment of seaweed reflects an extreme commitment to a single strategy. Where other fish rely on speed, schooling, or venom, the leafy sea dragon has invested entirely in resemblance. Its body has become an ecological statement, declaring that survival can be achieved not through confrontation, but through assimilation. In the shifting forests of kelp and seagrass, where motion is constant and outlines blur, the sea dragon stands as a testament to the power of becoming background. It does not merely hide among the leaves. It is, in every visible sense, one of them. There is no reef here, no rock to press against, no sand to bury into. The water column stretches in every direction, a three-dimensional expanse of blue fading into cobalt below and silver above. Sunlight pours down from the surface in diffuse shafts, scattering as it penetrates deeper, creating a gradient that predators and prey alike must constantly interpret. In this open pelagic world, silhouettes mean death. A fish seen from below against the brightness of the surface becomes a sharp outline, a dark cutout that announces its presence to anything hunting upward. A fish seen from above becomes a lighter patch against the gloom of depth. In such a habitat, there is nowhere to hide except within light itself. And suspended in this luminous void, a squid hovers almost imperceptibly, its body partially transparent, its shadow absent, its existence defined by the manipulation of photons rather than the occupation of space. The squid's first strategy is structural transparency. Its mantle, elongated and tapered, contains tissues with minimal pigmentation, allowing ambient light to pass through. Internal organs are arranged to reduce visual obstruction, and reflective structures, such as the digestive gland, are often shielded by silvered tissues that scatter light rather than absorb it. Transparency in marine organisms is a widespread adaptation among midwater species, particularly in larvae and small planktonic forms. However, complete transparency is physiologically difficult to achieve in larger, metabolically active animals. Muscles, nerves, and organs require density and vascularization that inevitably alter refractive indices relative to seawater. The squid navigates this constraint by combining partial transparency with active light production, a refinement that elevates concealment to an optical balancing act. Beneath its mantle, embedded within the skin, are photophores, specialized light-emitting organs that produce bioluminescence through chemical reactions. In many species inhabiting mesopelagic zones, these photophores are arranged along the ventral surface, facing downward. Their function is not to illuminate prey or signal mates, though in some contexts they may do so. Their primary role is counter-illumination, a strategy that cancels the squid's silhouette when viewed from below. Predators such as tuna, dolphins, and larger squid often patrol the dim layers beneath the surface, scanning upward for darker shapes against the residual glow of daylight filtering through water. A squid without counter-illumination would appear as a faint shadow. By emitting light that matches the intensity and wavelength of the surface illumination, the squid erases this contrast, effectively projecting brightness downward to equalize its outline. This process demands remarkable sensory integration. The squid must continuously assess the ambient light environment, which shifts with depth, time of day, cloud cover, and water turbidity. Specialized photoreceptors detect changes in light intensity, feeding information to neural circuits that modulate photophore output in real time. The bioluminescent reaction itself typically involves luciferin and luciferase, producing a cold light that consumes metabolic energy. The squid adjusts the brightness of this emission to mirror the gradient above, neither too bright to draw attention, nor too dim to leave a detectable shadow. It is an ongoing calibration, an invisible negotiation between organism and environment. The open ocean presents additional challenges beyond vertical illumination. Horizontal predators rely on motion detection and polarization sensitivity to locate prey. Squid possess complex eyes capable of detecting polarized light, an ability that enhances contrast in otherwise uniform blue fields. By perceiving polarized patterns reflected from other organisms, squid can discern subtle cues that betray presence. In turn, some species may adjust skin reflectance to minimize polarized signatures, further reducing detectability. This interplay of sensory adaptation and concealment underscores the arms race unfolding within the pelagic zone, where survival hinges on mastery of optical physics. Behavior complements physiology. Rather than swimming in erratic bursts, many midwater squid adopt measured, energy-efficient movements, maintaining position through gentle fin undulations and occasional jet propulsion. Rapid acceleration remains an option for escape, powered by expelling water through a siphon, but such movement is reserved for immediate threats. Continuous high-speed swimming would generate turbulence and visual disturbance, undermining transparency and counter-illumination. Thus, the squid balances readiness with restraint, conserving energy while minimizing perceptual cues. Predator-prey interactions in this environment are shaped by depth stratification. During daylight hours, many squid remain in deeper, dimmer layers, ascending toward the surface at night in a vertical migration that parallels that of zooplankton and small fish. This diel vertical migration represents one of the largest daily movements of biomass on the planet. By rising under cover of darkness, squid access abundant prey while reducing exposure to visually oriented predators. Counter-illumination remains relevant even in low light, as residual surface glow persists. The squid's ability to modulate light emission allows it to traverse gradients without becoming conspicuous. Feeding strategies reveal further integration of light manipulation. Some squid employ lures dangling bioluminescent appendages that attract small fish and crustaceans. Others rely on stealth, approaching prey with minimal disturbance before extending tentacles armed with suckers and hooks. The tentacular strike is rapid, drawing prey toward a beak capable of delivering precise cuts. In this moment of action, the squid transitions from invisible to lethal, then quickly returns to optical neutrality. The cycle underscores a fundamental principle. Invisibility in open water is not about permanent concealment, but about timing visibility on one's own terms. Thermoregulation in pelagic squid intersects with depth migration. Water temperature decreases with depth, influencing metabolic rate and enzymatic efficiency. By adjusting vertical position, squid can exploit temperature gradients to optimize digestion and energy expenditure. Warmer surface waters accelerate metabolic processes, facilitating rapid growth in juveniles. Cooler depths during daylight may reduce energy demands when feeding opportunities are limited. This thermal balancing act aligns with light-based concealment strategies, as depth selection simultaneously shapes optical conditions. Reproduction introduces another layer of complexity. Many squid species aggregate seasonally to spawn, forming transient concentrations in specific regions, influenced by currents and nutrient availability. Bioluminescence may play a role in mate recognition within these gatherings, though such signaling must remain controlled to avoid attracting predators. Egg masses are often deposited on substrates or released into the water column, depending on species. The transparency and counter-illumination that define adult survival give way to larval stages that rely heavily on transparency alone, drifting among plankton until growth permits more advanced optical strategies. Symbiotic relationships occasionally intersect with light production. Some squid species harbor bioluminescent bacteria within specialized light organs. These symbionts produce light through metabolic processes, reducing the squid's own energy investment in luminescence. The host regulates bacterial populations and light emission through mechanical shutters and chemical cues, maintaining control over brightness. This partnership exemplifies coevolution, where microbial and cephalopod lineages converge to solve the problem of open water invisibility. As dusk transitions to night, the ocean surface dims, and stars begin to reflect faintly above. In the water below, the gradient of brightness flattens, yet faint glows persist from distant cities, moonlight, or bioluminescent plankton. The squid continues its ascent, photophores adjusting output to match the new ambient conditions. Predators patrol beneath, scanning for discrepancies in the luminous field. The squid's body, semi-transparent and subtly illuminated, merges with the surrounding glow. Its shadow remains erased, its outline dissolved into the medium through which it moves. In a habitat devoid of physical cover, concealment demands mastery of light itself. The squid does not become rock or algae. It becomes brightness. By projecting light downward to cancel its own darkness, it manipulates the fundamental physics of visibility. Its survival depends on a dynamic equilibrium between emission and absorption, perception and deception. Within the open ocean, where every direction is exposed and every silhouette scrutinized, the squid demonstrates that invisibility is not an absence of form, but a precise alignment with the luminous architecture of water. On a shallow reef flat, where coral rubble and drifting algae create a restless mosaic of color and motion, a small crustacean moves with deliberate precision across the substrate. Its body is angular and compact, its legs jointed like mechanical arms, navigating a cluttered workshop. At first glance, it appears less like a crab and more like a fragment of reef debris nudged along by the tide. Patches of red algae cling to its back, delicate hydroids sway from its carapace, and bits of sponge protrude in irregular clusters. Only when the animal pauses and reaches upward with a slender claw does the illusion falter, revealing an architect at work. The decorator crab is not born fully camouflaged. It constructs its disguise, selecting and attaching living material to its own body, as though tailoring a garment from the reef itself. The behavior unfolds with methodical care, using specialized hooked setae, tiny hair-like structures covering its exoskeleton. The crab grips fragments of algae and sponge, trims them with its chelae, and presses them firmly into place. These hooked hairs function as natural Velcro, securing the chosen materials against the carapace. The result is a dynamic covering that evolves with the surrounding habitat. If the crab migrates from a patch of red algae to a field of green seagrass, it may gradually replace its adornments to better match the new environment. Unlike static camouflage, encoded solely through pigmentation, this strategy represents active camouflage, a behavioral extension of morphology that allows continuous adaptation to microhabitat variation. The ecological context in which decorator crabs operate is defined by intense predation pressure. Reef fishes, such as wrasses, triggerfish, and groupers, patrol the benthos, scanning for movement and recognizable outlines. In such visually complex habitats, detection often depends on identifying familiar shapes against a chaotic background. By breaking up its silhouette with living attachments, the decorator crab disrupts this recognition process. The added materials alter the contour of its body, creating asymmetry and irregularity that confound predators' visual systems. From a distance, the crab resembles an accidental cluster of algae rather than a mobile organism. The choice of materials is rarely random. Studies have shown that decorator crabs exhibit preferences for certain types of algae and sessile invertebrates, particularly those with chemical defenses. Some species deliberately select toxic sponges or stinging hydroids, gaining not only visual concealment, but also chemical protection. In these cases, camouflage becomes a form of symbiosis. The crab benefits from the defensive compounds of its chosen adornments, while the attached organisms gain mobility and access to new feeding grounds. This relationship blurs the line between decoration and partnership, embedding the crab within a network of ecological interactions that extend beyond mere appearance. The act of decorating carries energetic costs. Gathering materials requires time and exposure to predators, and the additional weight of attachments may reduce agility. Yet the benefits in reduced predation risk appear to outweigh these costs. Juvenile crabs, particularly vulnerable to predation, often exhibit more elaborate decoration than adults. As they grow and their exoskeleton thickens, the necessity for extensive coverage may diminish, though many individuals continue the practice throughout life. This ontogenetic variation suggests that camouflage intensity reflects life stage vulnerability and environmental conditions. Movement itself is adapted to preserve the illusion. Decorator crabs walk slowly, lifting their legs high and placing them carefully to minimize disturbance of the substrate. Rapid scuttling would betray their presence, so they rely on patience and incremental shifts. When threatened, they may freeze entirely, trusting their constructed disguise to withstand scrutiny. This stillness echoes the strategies of other ambush species, yet the crab's approach differs in its reliance on externally sourced materials rather than intrinsic morphological mimicry. Molting introduces a critical moment of vulnerability. As with all crustaceans, decorator crabs must periodically shed their exoskeleton to grow. During this process, they lose not only their hardened carapace, but also their attached camouflage. Immediately after molting, the crab's new exoskeleton is soft and unprotected, and it must rapidly redecorate to regain concealment. Observations reveal that crabs often reattach pieces of their previous decoration if available, suggesting a continuity of disguise across molts. The urgency of this behavior underscores the survival value of their constructed camouflage. Predator-prey interactions illustrate the effectiveness of this strategy. A triggerfish cruising above a coral outcrop may pass directly over a decorator crab without altering course. Only if the crab moves abruptly does the predator's attention sharpen. In controlled experiments, crabs deprived of decoration experience higher predation rates than those fully adorned, confirming the functional significance of their behavior. Interestingly, Some fish species appear capable of learning to recognize decorated crabs, indicating an evolutionary arms race between predator perception and prey deception. The materials chosen by decorator crabs also influence microhabitat structure by transporting fragments of algae and invertebrates across the reef. They contribute to the dispersal of these organisms. While the scale of this movement is small relative to ocean currents, it represents a localized vector for ecological connectivity. In areas where certain algae dominate, decorator crabs may inadvertently shape community composition by favoring specific species in their adornments. Thus, their camouflage behavior intersects with broader patterns of biodiversity and habitat maintenance. Thermoregulation plays a subtle role in this dynamic. In shallow reef flats exposed to intense sunlight, surface temperatures can rise significantly during low tide. The presence of attached algae may provide slight shading or moisture retention, buffering temperature fluctuations for the crab's exoskeleton. Although not the primary function of decoration, such incidental benefits highlight the multifunctional nature of adaptive traits. Evolution often refines behaviors that confer multiple advantages, even if secondary effects are modest. Reproductive cycles intersect with decoration in nuanced ways. During mating periods, males may reduce the density of their adornments to enhance mobility or visibility to potential mates. Conversely, females carrying eggs beneath their abdomens often maintain extensive camouflage, reducing predation risk during this vulnerable phase. The balance between concealment and reproductive signaling reflects competing selective pressures within the reef ecosystem. Sensory adaptations complement the crab's behavioral strategy. Compound eyes mounted on stalks provide a wide field of view, allowing the crab to monitor approaching threats without altering body position. Antennae sample chemical cues in the water, detecting predators or food sources. These sensory systems inform decisions about when to move, when to freeze, and when to gather new materials. The crab's world is one of constant evaluation, where each fragment of algae becomes both shield and statement. The decorator crab's approach to camouflage differs fundamentally from species whose disguise is encoded entirely within their own tissues. It demonstrates a form of environmental engineering at the individual level. By integrating external elements into its morphology, the crab transforms from passive recipient of evolutionary traits into active participant in its own concealment. This behavioral flexibility offers resilience in habitats subject to seasonal changes, storm disturbances, or shifts in species composition. As evening light softens the reef and plankton drift upward in the water column, the decorator crab continues its quiet labor. It reaches for a filament of red algae, trims it to size, and presses it against its carapace. The motion is deliberate, almost meticulous, yet entirely instinctive. Around it, fish weave through coral branches, unaware that the cluster of algae conceals a crustacean poised to vanish at the slightest disturbance. In this underwater landscape, camouflage is not merely a genetic inheritance, but a daily craft. The decorator crab stands as a testament to the ingenuity of evolutionary adaptation. Its invisibility is not fixed but curated, assembled piece by piece from the living reef. By customizing its appearance in response to shifting surroundings, it exemplifies a strategy that transcends static mimicry. The reef becomes both resource and refuge, and the crab's body becomes a moving fragment of its habitat. In a world defined by relentless predation and competition, survival sometimes depends not on speed or strength, but on the quiet artistry of blending in so completely that one ceases to be noticed at all. The tide turns over a shallow coral shelf, and the water begins to move with subtle insistence. Long ribbons of suspended sand drift in pale clouds above the reef, bending around coral heads and weaving through narrow channels between bommies. Sunlight fractures at the surface and descends in wavering bands that ripple across rock and seagrass alike. Within this constant distortion, a slender form advances, not in a straight line, but in a slow, rhythmic undulation that mirrors the water itself. From a distance, it resembles nothing more than a shifting shadow within the current, a darker strand caught in the flow. Only when the head rises slightly above the surface to take a breath does the pattern resolve into scales and a narrow skull. The sea snake has not concealed itself behind coral or buried into sand. It has aligned its body so precisely with the movement of water that it becomes indistinguishable from the distortion of light and tide. Sea snakes inhabit tropical and subtropical coastal waters across the Indo-Pacific, occupying environments that range from coral reefs and mangroves to estuaries and open lagoons. Unlike terrestrial snakes that rely heavily on static background matching, many sea snakes move continuously through fluid habitats where patterns and boundaries are perpetually shifting. Their camouflage does not depend solely on color but on the dynamic interplay between banding patterns and the optical turbulence of shallow seas. Alternating bands of black and white or muted browns and grays fragment the animal's outline, creating discontinuities that disrupt the perception of a coherent body. In waters where sunlight flickers through surface waves, these patterns merge with caustic light, transforming the snake into a living extension of the current. The effectiveness of this strategy becomes apparent when observing prey species. Small reef fish rely on rapid visual assessment to detect predators, scanning for recognizable silhouettes and sudden directional changes. A sea snake advancing through a reef does not present a stable outline. Its sinuous motion blends with the swaying of soft corals and the oscillation of seagrass blades. Each curve of its body overlaps with shifting shadows cast by moving water. The predator's form becomes an extension of environmental motion, and prey often register only a vague fluctuation in brightness rather than a distinct threat. This perceptual ambiguity buys crucial seconds, time enough for the snake to approach striking distance. Physiologically, sea snakes are highly specialized for aquatic life. Their tails are laterally compressed, forming paddle-like structures that generate propulsion with minimal lateral displacement. The rest of the body remains relatively cylindrical, maintaining hydrodynamic efficiency. Valved nostrils seal during submersion, and a single elongated lung extends through much of the body cavity, aiding buoyancy control. By adjusting lung volume, sea snakes can maintain neutral buoyancy, hovering within the water column without constant muscular effort. This buoyancy control allows them to drift subtly with currents, reinforcing the illusion of being part of the water's motion rather than an independent organism. Venom adds another layer to their ecological strategy. Sea snakes possess potent neurotoxins delivered through fixed front fangs, primarily targeting fish and eels. Their venom is among the most effective in the marine environment, evolved to immobilize agile prey quickly. Yet sea snakes rarely display overt aggression toward larger animals, including humans. Their camouflage through motion serves not only hunting, but also defense, reducing encounters with predators such as sharks and large reef fish. In many instances, potential predators fail to recognize the snake until it has already passed through their field of vision, minimizing confrontation. The interplay between motion and pattern is particularly striking in shallow mangrove channels, where tannin-stained waters create a dim, amber haze. Here, light penetration is limited, and visual acuity decreases. The banded bodies of sea snakes dissolve into alternating streaks that mimic submerged roots and drifting debris. When currents accelerate, suspended particles create a flickering veil that further obscures outlines. In such conditions, the snake's progress becomes nearly impossible to track, its path merging with the organic geometry of the habitat. This is not invisibility through stillness, but through synchronized movement. Thermoregulation in sea snakes intersects with habitat selection and daily activity patterns. As ectotherms, their body temperature fluctuates with water temperature, influencing metabolic rate and digestive efficiency. Many species engage in surface basking behaviors, raising portions of their bodies above water to absorb solar radiation. This exposure is brief and calculated, balancing the need for warmth with vulnerability to aerial predators. Once warmed, they submerge and resume hunting in cooler layers. The vertical distribution of sea snakes often mirrors temperature gradients and prey abundance, illustrating the integration of physiological and ecological constraints. Reproductive strategies also reflect adaptation to a fully aquatic lifestyle. Most sea snakes are viviparous, giving birth to live young in the water rather than returning to land to lay eggs. This reproductive mode eliminates the need for terrestrial nesting sites, allowing populations to remain entirely marine. Neonates are born fully formed and capable of independent hunting, their patterns already aligned with environmental distortion. The continuity of banding across generations underscores the evolutionary stability of this camouflage strategy. Predator-prey interactions reveal moments of startling precision as a sea snake approaches a cluster of juvenile fish sheltering among coral branches. Its head weaves subtly, sampling chemical cues with a forked tongue that flicks briefly into the water. The sensory organ, known as the Jacobson's organ, interprets these cues, guiding the snake toward concentrations of prey. When within range, the strike is swift and localized, the body anchoring momentarily against coral to generate leverage. The prey's recognition of danger often occurs too late, as the snake's approach was masked by environmental motion until the final instant. Interestingly, some reef fish have evolved partial tolerance or avoidance strategies in response to sea snake predation. Observations suggest that certain species alter schooling behavior when sea snakes are present, increasing spacing or shifting depth. This behavioral plasticity demonstrates the ongoing evolutionary dialogue between predator and prey, where camouflage drives adaptation in detection mechanisms. Yet even in such cases, the sea snake's ability to blend into distortion complicates effective avoidance. Symbiotic relationships occasionally intersect with this ecological web. Cleaner fish may approach sea snakes to remove parasites, a risky interaction given the snake's predatory capacity. During such encounters, the snake often remains motionless, allowing cleaning to proceed. Its banded pattern remains static, but the absence of aggressive movement maintains a temporary détente. These interactions highlight the nuanced balance between predation and cooperation within reef ecosystems. As evening descends and light fades, the dynamics shift once more. Surface reflections soften, and the gradient between brightness and shadow flattens. The sea snake's banding becomes less pronounced, merging into the darkening water. No longer relying on high-contrast fragmentation, it benefits from overall reduction in visibility. Predators that hunt by sight become less effective, and the snake's movements grow more deliberate. In these twilight hours, It navigates channels between coral heads with quiet confidence, its presence sensed more by subtle pressure changes than by sight. The sea snake's mastery lies not in vanishing against a static backdrop, but in becoming inseparable from motion itself. In habitats defined by fluidity and light distortion, it adopts the characteristics of water, aligning its body with currents and breaking its outline into rhythmic segments. Its camouflage operates on the principle that recognition requires stability. By denying observers a stable form, it remains unrecognized. The threat it represents is not concealed behind rock or sand, but embedded within the very patterns of moving water. By the time the brain assembles the fragments into a coherent predator, the silent shadow has already passed or struck. A narrow mouth gapes in the dim green light, lined with recurved teeth that catch and scatter the sun's fractured beams. The jaws open and close rhythmically, revealing a second, deeper set of bones, sliding forward within the throat, as if the reef itself were chewing. The eye, small and unblinking, appears fixed in a permanent glare. From this perspective, the creature seems exposed, almost reckless in its display. Yet as the camera eases backward, the monstrous face begins to dissolve. The skin's mottled browns, olives, and creams align seamlessly with the limestone walls. The elongated body disappears into a fissure no wider than a human wrist. What moments ago looked like a predator on display is revealed as architecture, a living extension of the coral reef's fractures and shadowed cavities. The moray eel does not patrol its territory in open water. It inhabits the reef's negative space so completely that predator and habitat become indistinguishable. Coral reefs are not solid fortresses, but labyrinths of tunnels, crevices, and chambers formed by centuries of coral growth and bioerosion. Parrotfish grind coral skeleton into sand. Boring sponges hollow out chambers, and the relentless push of waves fractures older structures into intricate passageways. These voids are ecological highways and shelters used by shrimp, crabs, small fish, and countless invertebrates seeking refuge from open water predators. The moray eel exploits this architecture with extraordinary precision. Its body is elongated and laterally compressed, lacking the prominent pectoral fins seen in most fish, which allows it to thread through narrow spaces with minimal resistance. The absence of scales reduces friction against rock surfaces, while a thick mucus coating protects its skin from abrasion and microbial infection. Rather than adapting to the open water column, the moray has refined itself to inhabit the reef's cracks, where space is limited and visibility is fragmented. The fearsome gape that defines its silhouette serves a physiological purpose beyond intimidation. Moray eels ventilate their gills by opening and closing their mouths, pumping water across respiratory surfaces in the absence of large gill openings or opercular flaps. What appears to be a perpetual threat display is, in reality, a necessity of respiration. Yet this involuntary motion has an ecological side effect. Prey species often misinterpret it as aggressive readiness. The psychological dimension of predation is subtle here. Small fish scanning the reef may hesitate when confronted with a cavern containing a set of teeth, even if they cannot discern the rest of the body. That hesitation can alter movement patterns, influencing how fish distribute themselves among crevices and thereby shaping microhabitat usage within the reef. As daylight filters through the water column, the reef becomes a mosaic of brightness and shadow. Hard corals cast sharp silhouettes, while soft corals sway in slow arcs. Within this interplay of light, the moray's skin pattern, often a complex marbling of irregular blotches, mirrors the reef's textures. The pigmentation breaks up the continuity of its body, creating the illusion of depth and irregularity. From even a short distance, the exposed head appears as nothing more than a darker pocket within stone. The rest of the eel remains concealed, coiled in loops that follow the contour of the crevice. Its strategy is not pursuit, but integration. It waits as an element of structure, transforming a static refuge into a lethal threshold. Predation unfolds not in dramatic chases, but in compressed, decisive bursts. A small wrasse, emboldened by familiarity with its surroundings, slips into a crevice to forage for crustaceans. The eel remains motionless, its sensory world defined less by sight and more by chemical gradients. Moray eels possess well-developed olfactory organs, capable of detecting minute traces of amino acids released by injured or stressed prey. Water moving through the crevice carries these signals directly to the eel's nostrils. When the wrasse crosses an invisible boundary, often mere centimeters from the eel's jaws, the strike is instantaneous. The elongated body anchors itself within the rock, generating leverage, while the jaws snap forward. In the same fluid motion, a second set of jaws, known as pharyngeal jaws, thrust from the throat to grasp the prey and pull it inward. This mechanism, rare among fishes, compensates for the limited suction, feeding capacity inherent to a narrow head adapted for crevices. Evolution has traded broad mouths for interior weaponry. The relationship between moray eels and other reef predators introduces an unexpected layer of ecological complexity. In certain regions, groupers and morays engage in cooperative hunting. A grouper may signal a concealed moray by shaking its head near a crevice. The eel, responsive to the movement, emerges and begins probing cavities inaccessible to the larger fish. Prey, attempting to flee the eel's intrusion, often bolt into open water, where the grouper intercepts them. Conversely, prey escaping the grouper's pursuit may retreat into crevices, only to encounter the waiting eel. This partnership is not born of affection, but of mutual benefit, a calculated alliance between species occupying complementary hunting niches. It underscores how structural specialization can integrate into broader predator networks within the reef ecosystem. Thermoregulation plays a subtler role in the moray's daily rhythm. Reef waters fluctuate in temperature depending on depth, tidal movement, and solar radiation. As ectotherms, morays adjust their activity levels according to ambient conditions. During cooler periods, they may remain deeper within crevices, conserving energy and reducing metabolic demands. As water warms, digestion accelerates, and hunting activity increases. The timing of feeding often aligns with crepuscular periods, dawn and dusk, when prey species shift between shelter and open foraging grounds. In these transitional moments, visibility changes rapidly, enhancing the eel's advantage as a stationary predator embedded within structure. Symbiotic interactions further integrate the moray into reef life. Cleaner shrimp and small fish approach the eel's open mouth to remove parasites and dead tissue. The eel's tolerance of such intrusions appears remarkable given its predatory nature, yet the benefits of parasite removal outweigh the potential meal. These cleaning stations often form in predictable locations within the reef, turning certain crevices into hubs of mutualistic exchange. The eel's fearsome visage becomes, briefly, an anchor point for ecological cooperation, illustrating that even apex micropredators are embedded in networks of reciprocal relationships. The structural role of the moray extends beyond predation. By occupying crevices and exerting predation pressure on smaller fishes and invertebrates, morays influence species composition within the reef's hidden chambers. Their presence can deter overpopulation of certain prey species, indirectly affecting algal growth and coral health. Predation is not merely an act of consumption, but a regulatory force that maintains balance within confined habitats. In reefs where moray populations decline, shifts in prey abundance can cascade through trophic levels, altering the fine equilibrium that supports coral resilience. At night, the transformation becomes more apparent. As darkness envelops the reef, diurnal fishes retreat and nocturnal organisms emerge. The moray, no longer confined to a single crevice, may extend much of its body into open water under cover of reduced visibility. Its sinuous movement through coral corridors remains deliberate, guided by scent trails rather than sight. The same body that merges seamlessly with stone during daylight now navigates actively among structures, exploring new cavities. Yet even in motion, it remains tethered to the reef's architecture, rarely venturing far into open water. The reef is not merely habitat, but identity. The terrifying face that first commands attention is therefore only a fragment of the story. What seems like exposure is, in fact, concealment perfected through structural mimicry and behavioral restraint. The moray eel does not rely on speed or distance pursuit. It has reshaped itself to become an extension of the reef's interior, converting shelter into ambush and shadow into strategy. Within the living limestone labyrinth, predator and habitat converge so completely that prey often fail to perceive the boundary between safety and danger. The reef's crevices are not empty spaces awaiting occupation. They are inhabited, animated, and armed. The moray eel is less a hunter moving through coral than a living crevice, a silent component of the reef's architecture that waits patiently for life to brush too close against stone. The first impression is unmistakable. A dark sphere bristling with spines, radiating danger in every direction. It sits wedged between coral heads, its silhouette seemingly impossible to miss. Hundreds of needle-like projections extend outward in rigid symmetry, catching stray beams of sunlight and casting thin, shifting shadows across the reef floor. At close range, the sea urchin appears almost theatrical in its defensiveness, as though evolution exaggerated every warning signal. Yet as the camera drifts backward and the surrounding reef comes into view, the illusion begins to unravel. What seemed obvious dissolves into a field of equally jagged textures: branching corals, filamentous algae, shattered limestone, the spiny arms of brittle stars, and the needle forests of neighboring urchins. In this crowded, chaotic architecture, the sea urchin does not hide by resembling a single object. It disappears by overwhelming the eye's ability to organize shapes. Its camouflage is not subtlety, but confusion. Coral reefs are environments defined by structural complexity. Unlike sandy plains or open water columns, reefs present a three-dimensional mosaic of hard skeletons, soft tissues, shadows, and overlapping organisms. Every square meter can contain thousands of individual invertebrates and fish, each contributing to a visual field saturated with edges and angles. In such conditions, the human brain and the visual systems of reef predators struggles to distinguish discrete forms from background noise. This phenomenon, known in ecological research as disruptive patterning and background matching through complexity, does not require perfect color mimicry. Instead, It relies on breaking the continuity of recognizable outlines. The sea urchin's radial symmetry and protruding spines fracture its own silhouette into so many intersecting lines that the eye fails to isolate it as a single, cohesive organism. The spines themselves vary widely in length, thickness, and pigmentation, depending on species and habitat. In some tropical reefs, long-spined urchins, such as those in the genus Diadema, carry slender black spines that extend outward like an inverted forest. In other species, spines are shorter, thicker, and mottled in browns, purples, or greens, blending with coralline algae and encrusting sponges. These variations are not random. Spine morphology influences both predator deterrence and hydrodynamics. Longer spines create a larger apparent body size, increasing the difficulty for gape-limited predators, such as triggerfish, to access the central test, or shell. At the same time, the flexible articulation of spines allows the urchin to withstand wave energy, bending slightly rather than snapping under force. In high-energy reef zones, where surge is constant, the interplay between spine flexibility and skeletal rigidity becomes critical to survival. From a distance, however, the sea urchin's spines do not read as a unified defense. They merge visually with the reef's chaotic geometry. Branching corals produce similarly fine projections. Algal filaments sway in comparable patterns. Even the shadows cast by rippled surface light create radiating lines that mimic the urchin's outline. The result is a perceptual overload. Predatory fish scanning for prey rely on detecting coherent shapes and consistent motion. The sea urchin offers neither. It presents instead a tangle of lines embedded within an already tangled world. In dense aggregations, where dozens of urchins cluster together in crevices, the effect intensifies. Individual boundaries vanish entirely, replaced by a collective thicket that appears as a structural feature rather than a biological one. This camouflage by confusion becomes particularly effective in twilight conditions. As daylight fades, contrast decreases, and the visual acuity of diurnal predators declines. In these moments, the reef's surface becomes a uniform patchwork of shadow and low light. The urchin's spines, once sharp and defined, soften into indistinct gradients. No longer conspicuous spheres, they become ambiguous voids among coral rubble. For predators that rely on rapid identification, hesitation can mean lost opportunity. The urchin's strategy exploits this hesitation, turning apparent visibility into practical invisibility. Beyond visual defense, the sea urchin's anatomy reveals a complex integration of sensory and locomotor adaptations. Beneath the spines lies the rigid test, composed of interlocking calcium carbonate plates. Small tube feet protrude through pores in this shell, operated by a hydraulic water vascular system unique to echinoderms. These tube feet adhere to rock surfaces with remarkable strength, allowing the urchin to anchor itself against currents or reposition slowly across the substrate. Movement is deliberate, almost imperceptible at human timescales. Yet over hours and days, urchins can traverse significant distances in search of food. Their diet primarily consists of algae scraped from rock surfaces using a specialized feeding apparatus known as Aristotle's lantern, a complex arrangement of 5 calcareous teeth capable of grinding hard substrates. In performing this grazing function, sea urchins act as regulators of algal growth, preventing macroalgae from overwhelming coral colonies and maintaining open surfaces for coral larvae to settle. This ecological role introduces a paradox. While individual urchins seek concealment through confusion, their collective grazing shapes the reef's physical appearance. In areas Where urchin populations thrive, algae remain cropped short, exposing pale limestone and creating visually open spaces. Conversely, where urchins decline, often due to disease outbreaks or overfishing of their predators, algal overgrowth can smother coral reefs, reducing biodiversity. Thus, the organism that vanishes into chaos contributes to maintaining the structural diversity that enables its own camouflage. The reef's complexity is both shield and product of urchin activity. Predator-prey interactions around sea urchins are nuanced. Certain fish, such as triggerfish and wrasses, have evolved specialized behaviors to overcome the spiny defense. A triggerfish may blow jets of water at an urchin, attempting to flip it and expose the softer underside. This maneuver requires precision and persistence, as the urchin responds by reorienting itself using coordinated spine movements and tube feet. The spines can pivot toward a perceived threat, forming a directional barrier. In some species, venomous pedicellariae, tiny pincer-like appendages between spines, deliver additional deterrence. Yet even in these confrontations, the surrounding reef complicates the scene. The visual clutter makes it difficult for predators to isolate a single target among many similar shapes. The battle becomes not just a test of strength, but of perception. Symbiotic relationships add further layers to the illusion. Small fish, such as cardinal fish and juvenile damselfish, often shelter among urchin spines, gaining protection from larger predators. Their presence introduces motion within the spiny mass. Yet this movement can paradoxically enhance camouflage. The flickering of small bodies among spines resembles the general activity of the reef, blending into the background noise of constant minor motion. In some cases, crabs or shrimp reside within the urchin's protective canopy, forming microhabitats that expand the reef's biodiversity. The urchin becomes not merely an individual organism, but a structural refuge within the ecosystem. Thermoregulation in sea urchins, while less dramatic than in mobile predators, remains vital. As ectothermic invertebrates, their metabolic processes are governed by ambient water temperature. In shallow reefs exposed to intense sunlight, temperatures can fluctuate significantly over the course of a day. Urchins often retreat deeper into crevices during peak heat, reducing exposure and minimizing desiccation risk during extreme low tides. In temperate zones, seasonal temperature shifts influence reproductive cycles, with spawning events synchronized to environmental cues such as water temperature and lunar phase. Gametes released into the water column depend on currents for fertilization, illustrating how even sedentary organisms participate in dynamic, large-scale ecological processes. Perhaps the most surprising aspect of the sea urchin's camouflage is its psychological effect. To human observers, the spiny sphere appears aggressive and conspicuous. Yet in the context of a reef saturated with spikes, ridges, and branching forms, the urchin's geometry becomes statistically unremarkable. Visual systems, whether human or piscine, evolved to detect anomalies, shapes that deviate from background expectations. The sea urchin's genius lies in matching the reef's statistical chaos so closely that it ceases to be an anomaly. It is not invisible because it blends into a uniform color field. It is invisible because it contributes to and reinforces complexity itself. As currents shift and schools of fish sweep across the reef, the sea urchin remains anchored, its spines swaying slightly with the movement of water. It neither pursues nor flees in dramatic arcs. Its survival depends on embedding itself so thoroughly within structural confusion that predators overlook it until contact is unavoidable. Even then, the spines serve as both barrier and warning, converting confusion into defense. The reef's chaotic beauty, its overlapping textures and relentless intricacy, provides the stage upon which the current moves first. Long strands of seagrass bend in a slow, synchronized arc, their narrow blades tilting toward the sandy bottom before rising again as the surge reverses. Suspended particles shimmer in the filtered light, tracing invisible loops through the water column. Within this submerged meadow, a slender shape hangs vertically, tethered by a coiled tail to a single blade. At a glance, it appears to be a frayed fragment of vegetation, perhaps a piece of drifting algae caught among the stems. Only when the camera settles and the water stills for a moment does the outline clarify into the profile of a horse-like head and a segmented body plated in bony rings. The seahorse does not flinch or dart away. It continues to sway, its motion so perfectly synchronized with the surrounding plants that it seems governed by the same unseen hand guiding the grass. The deception is not in altering its form, but in surrendering to rhythm. Seahorses inhabit coastal environments where structural vegetation dominates. Seagrass beds, mangrove roots, and coral branches. These habitats are defined by constant movement driven by tides, wind, and wave energy. Any object that remains rigid against this motion stands out immediately. Evolution has shaped the seahorse into a master of compliance. Its body, encased in a rigid exoskeleton of interlocking bony plates, might appear inflexible at first glance. Yet this rigidity serves as an anchor for subtle adjustments. The tail, prehensile and muscular, coils securely around stems or coral branches, allowing the animal to pivot gently as currents pass. Rather than resisting water flow, the seahorse yields to it, converting what could expose it into a camouflage mechanism based on timing and amplitude of sway. The resemblance to plant structure is precise. Many seahorse species display elongated snouts and coronets that mimic the irregularities of leaves or algal fronds. Some carry filamentous skin appendages that resemble epiphytic growths clinging to vegetation. Pigmentation varies widely, often matching the dominant tones of the surrounding habitat: greens and yellows in lush meadows, browns and mottled patterns in mangroves or coral rubble. Unlike cephalopods, seahorses do not rapidly change color to match new backgrounds. Their concealment depends on selecting and remaining within environments that align with their morphology. This strategy emphasizes habitat fidelity over versatility, an evolutionary choice that trades adaptability for precision. Predation pressure in these shallow ecosystems is intense. Juvenile fish, crustaceans, and even other seahorses fall prey to larger reef inhabitants. Seahorses, slow swimmers by design, cannot rely on speed to escape. Their dorsal fin beats rapidly, up to several dozen oscillations per second, but propulsion remains modest. Instead, they minimize detection altogether. The key lies in motionlessness, punctuated by controlled drift. When currents strengthen, the seahorse's body angles slightly to reduce drag, while the tail tightens its grip. When the water calms, the animal remains still, its head aligned with vertical stems. Predatory fish, scanning the meadow for irregularities, often overlook these stationary figures, interpreting them as part of the vegetative matrix. Feeding behavior reinforces this stillness strategy. Seahorses are ambush micropredators, targeting small crustaceans such as copepods and mysids. Their elongated snouts function as precise suction devices. When prey drifts within striking range, the seahorse pivots its head in a rapid upward motion, creating negative pressure that draws the prey into its mouth. This strike occurs in a fraction of a second, powered by elastic recoil stored in neck tendons. The body itself remains largely stationary, anchored to vegetation. Such efficiency minimizes disturbance to the surrounding plants, preventing sudden movements that might betray the animal's presence. Each feeding event integrates seamlessly into the meadow's rhythm. Thermoregulation in seahorses reflects the constraints of shallow coastal habitats. Water temperatures can fluctuate daily and seasonally, influencing metabolic rates and reproductive timing. As ectotherms, seahorses adjust activity levels according to temperature, feeding more actively in warmer conditions when metabolic processes accelerate. In cooler waters, digestion slows, and individuals may cluster in sheltered zones to conserve energy. Their limited swimming capability reinforces dependence on microhabitats that provide both food availability and thermal stability. In dense seagrass beds, the canopy moderates temperature extremes, creating microclimates that buffer sudden environmental changes. Reproductive biology introduces one of the most unusual adaptations in marine life: male pregnancy. During courtship, which may involve synchronized swaying and color intensification, the female deposits eggs into a brood pouch located on the male's abdomen. Fertilization occurs within this pouch, where embryos develop protected from external threats. The male regulates salinity within the pouch, gradually acclimating, developing young to seawater conditions. This internal incubation reduces egg predation and increases offspring survival, albeit at the cost of limited brood size. When the young are released, miniature replicas of adults drift briefly before seeking their own holdfasts among vegetation. From the earliest stages, survival hinges on mastering stillness within motion. Symbiotic relationships further illustrate the seahorse's integration into its environment. Small invertebrates sometimes settle on their bodies, adding texture that enhances camouflage. In some cases, seahorses position themselves near colonies of hydroids or soft corals, where the combined complexity of branching organisms amplifies concealment. The presence of these additional textures blurs the boundary between animal and plant even further. Predators encountering such scenes must distinguish between living prey and static structure, a task complicated by the uniform sway of all elements. The ecological role of seahorses extends beyond individual survival. By preying on planktonic crustaceans, they influence microfaunal populations within seagrass ecosystems. These meadows serve as nurseries for numerous fish species, and the balance of small invertebrates affects nutrient cycling and algal growth. Seahorses, though diminutive and seemingly fragile, participate in regulating these delicate systems. Their reliance on intact vegetation makes them sensitive indicators of habitat health. Declines in seagrass due to pollution or coastal development often correspond with reductions in seahorse populations, underscoring their dependence on structural complexity. Migration patterns in seahorses are limited compared to pelagic fish. Many individuals exhibit strong site fidelity, remaining within a small home range for extended periods. However, seasonal changes or habitat disturbances can prompt relocation. Movement occurs slowly and cautiously, often at night when visual predators are less active. Even during transit, the seahorse seeks intermittent anchor points, progressing in stages rather than continuous swimming. This methodical relocation reflects a life history strategy centered on minimizing exposure rather than maximizing speed. Observing a seahorse over several tidal cycles reveals subtle behavioral nuances. As the tide rises, bringing cooler offshore water, feeding intensity may increase with the influx of plankton. During slack tide, when water movement diminishes, the animal's sway becomes almost imperceptible. In these moments, it appears carved from the meadow itself, a stationary ornament in a living landscape. The illusion depends on restraint. Any abrupt movement would fracture the visual harmony, drawing attention to the imposter among stems. In the broader context of evolutionary strategy, the seahorse exemplifies a path less defined by aggression or agility and more by synchronization, where other predators refine speed, venom, or brute strength. The seahorse refines timing and compliance. It has evolved to match not just the appearance, but the cadence of its surroundings. This alignment extends beyond camouflage into behavioral ecology. Its daily rhythms mirror tidal cycles. Its reproductive schedule aligns with seasonal currents. Its feeding strategy depends on the passive delivery of prey by water flow. Every aspect of its biology converges on the principle of harmonizing with environmental motion. As dusk approaches and the meadow dims, the seahorse remains tethered, its silhouette merging with darkening blades. The current continues its steady pulse, bending grass and animal alike in unison. There is no dramatic chase, no overt display of power. Survival unfolds in measured stillness, in the discipline of remaining indistinguishable within a field of perpetual movement. The seahorse's success lies not in transforming itself to match every new background, but in selecting a rhythm and inhabiting it so completely that it ceases to be separate from the ecosystem. In a world where detection often determines fate, it survives by becoming part of the current's choreography, an organism whose greatest strength is its refusal to break the pattern. The seabed appears undisturbed, a broad expanse of pale sand rippled by the steady rhythm of tidal flow. Each ridge casts a faint shadow in the angled light, creating a repeating pattern that stretches toward the horizon of the shallow lagoon. Small gobies hover just above the substrate, darting from ripple to ripple in search of tiny crustaceans. A hermit crab drags its borrowed shell across the grains, leaving a temporary track that the next pulse of current will erase. At first glance, there is no sign of a predator here. The surface is uniform, quiet, and open. Then, almost imperceptibly, two raised spiracles widen and contract. A pair of eyes, positioned atop what seemed like empty sand, rotate slightly. The phantom reveals itself, not by moving forward, but by breathing. The stingray lies buried beneath a thin veneer of sediment, its flattened body pressed into the seabed with such precision that only the dorsal surface remains near exposure. Using undulating motions of its pectoral fins, it has drawn sand over its back, directing a fine stream of grains across its disc until its outline dissolves into the surrounding texture. The tail extends backward, partially concealed along the ripple trough. From above Wiki, the only visible markers are the protruding eyes and spiracles, structures designed to function even when the rest of the body is submerged beneath sand. The illusion is not merely color matching, it is architectural erasure. The stingray has reduced its visible presence to the smallest possible surface area, consistent with respiration and vigilance. Shallow coastal habitats, where many stingrays reside, are defined by constant reshaping. Waves, tides, and wind-driven currents rearrange sediments daily, creating dunes and depressions that shift over time. In such dynamic environments, rigid camouflage strategies would fail quickly. The stingray's approach relies on behavior integrated with morphology. Its dorso-ventrally flattened body distributes weight evenly, preventing it from sinking too deeply into soft sediment while still allowing partial burial. The skin, often modeled in tans, grays, or speckled patterns, mirrors the granular variability of sand. Some species display intricate reticulations that break up the disk's perimeter, ensuring that even when partially exposed, the eye struggles to trace a continuous edge. Burial is not passive. Upon settling, the stingray performs a deliberate sequence of movements. The edges of its disk lift and fold in coordinated waves, generating localized currents that lift sand from beneath and redirect it over the dorsal surface. This self-burial process can occur in seconds, transforming a conspicuous fish into a low-profile mound, indistinguishable from natural seabed irregularities. Once covered, the stingray remains still, relying on sensory systems attuned to subtle vibrations and electrical signals. Embedded within its snout are electroreceptors known as ampullae of Lorenzini, capable of detecting minute electrical fields produced by the muscle contractions of buried prey. Even beneath layers of sand, the ray can perceive the hidden movements of worms, mollusks, and small fish. Respiration under sediment requires adaptation. Unlike many fish that draw water through mouths positioned anteriorly, stingrays intake water through spiracles located behind the eyes. These openings remain above the sand, pulling water downward across the gills and expelling it ventrally. This arrangement allows the ray to breathe while its mouth and gill slits remain buried. The separation of feeding and respiratory pathways reduces the intake of sediment during both processes, a crucial refinement for an organism that spends extended periods beneath sand. What appears to be a simple resting posture is in fact a carefully engineered physiological stance. Predation unfolds as a matter of proximity rather than pursuit. When a small crab ventures across the ripple field, probing the substrate for detritus, it unknowingly passes within range of the concealed ray. The stingray senses the crab's muscular impulses and perhaps even the faint electrical disturbances generated by its nervous system. At the critical moment, the ray erupts upward in a rapid upward thrust, its disc lifting sand in a brief cloud. The mouth opens on the ventral side, creating suction that draws the crab inward before it can react. The entire event may last less than a second. Sand resettles almost immediately, restoring the seabed's deceptive calm. To an observer scanning the horizon, nothing has changed. The stingray's defensive apparatus is equally integrated into its low-profile strategy. Along the dorsal surface of the tail lies one or more serrated spines, composed of dentine and coated in venomous tissue. These barbs are not offensive weapons used in hunting, but deterrents activated when the animal is threatened. The ray prefers immobility and avoidance. When approached too closely by a potential predator or an unwary wader, it may lash its tail in a swift arc, delivering a puncture that injects venom and deters further contact. Yet this defense is secondary to concealment. The ray's first line of survival is partial disappearance. Thermoregulation in shallow water stingrays reflects the interplay between behavior and environment. Sandy flats can experience dramatic temperature fluctuations throughout the day, particularly in tropical lagoons, where midday sun warms shallow water rapidly. By burying into cooler sediment layers, rays may buffer themselves against surface heat. Conversely, in cooler conditions, remaining partially exposed allows passive absorption of solar radiation. Some species undertake seasonal migrations to deeper waters during colder months, adjusting distribution patterns in response to thermal thresholds that influence metabolism and reproductive timing. These movements, though less dramatic than oceanic migrations, underscore the sensitivity of benthic organisms to climatic variation. The stingray's ecological role extends beyond predation. By disturbing sediment during feeding and burial, rays contribute to bioturbation, the mixing of seabed layers that influences nutrient cycling and oxygen penetration. This activity affects microbial communities and invertebrate populations, subtly reshaping the benthic ecosystem. In areas where ray populations are abundant, patterns of sediment disturbance become visible from above as overlapping pits and mounds, evidence of continuous interaction between predator and substrate. The sand phantom does not merely inhabit the seabed, it engineers it. Interactions with other species reveal additional complexity. Certain small fish follow feeding stingrays, capitalizing on prey items exposed during sediment disturbance. This opportunistic association demonstrates how one species' hunting behavior creates feeding opportunities for others. Conversely, larger predators, such as sharks, may prey upon stingrays, flipping them to access the unprotected underside. In these encounters, burial may provide insufficient protection, and the ray relies on sudden bursts of swimming powered by powerful pectoral fin strokes. Yet even in flight, the stingray's flattened body enables rapid gliding close to the seabed, reducing detection from above. As dusk settles over the lagoon, light levels diminish, and the contrast between sand ridges softens. The stingray may shift position, gliding silently to a new depression before settling again. Its movements are fluid, almost weightless, yet always return to stillness. The strategy remains consistent: reduce visibility, minimize exposure, rely on the environment to conceal. The eyes remain vigilant, rotating independently to monitor both horizon and overhead space. Even when half-buried, awareness persists. What defines the sand phantom is not invisibility in the conventional sense, but subtraction. The stingray subtracts itself from the visible field until only essential components remain. Eyes and spiracles protrude like minor imperfections in an otherwise continuous plane. The rest becomes indistinguishable from mineral substrate. In a landscape shaped by erosion and flow, this partial disappearance proves more effective than elaborate patterning. The seabed, seemingly empty at first glance, is in fact inhabited by organisms that master the art of absence. Standing at the water's edge and gazing across the shallow flats, one might see only wind-rippled sand and the occasional flicker of fish. Yet beneath that calm surface, forms lie in wait, embedded within the very structure of the habitat. The stingray exemplifies a survival strategy rooted in integration with sediment, physiology aligned with burial, and sensory systems attuned to hidden life beneath the sand. Its presence is felt not through spectacle, but through the subtle distortion of a landscape that conceals it. The phantom does not vanish into background through color alone. It becomes background, surrendering its outline to the seabed until recognition requires careful scrutiny. In the quiet geometry of shallow water, disappearance is an active choice, and the stingray executes it with disciplined precision. The reef does not sleep, but it changes character. As sunlight drains from the water column and the brilliant palette of corals dulls into muted blues and grays, the hierarchy of hunters rearranges itself. Diurnal grazers retreat into crevices, while nocturnal predators, moray eels, reef sharks, large groupers, begin to patrol with slow, deliberate intent. In the half-light beneath an overhang, a parrotfish wedges itself into a narrow recess in the coral. Its vibrant daytime colors, once a declaration of presence among branching corals, now appear conspicuous in the fading glow. Yet instead of relying solely on shadow, or rock for concealment, the fish begins a subtler transformation. Its mouth, built for scraping limestone and algae from coral skeletons, now secretes a translucent stream of mucus. The material spreads outward from the gill openings and lips, enveloping the body in a thin gelatinous sheath. Within minutes, the parrotfish is suspended inside a cocoon of its own making, sealed from the surrounding water by a nearly invisible membrane. The disguise is not visual flamboyance, but chemical silence. Parrotfish are primarily herbivorous reef inhabitants, equipped with fused teeth that form a beak capable of grinding coral substrate to extract microscopic algae. Their feeding activity during daylight hours contributes significantly to reef ecology, converting coral skeleton into fine sand and preventing algal overgrowth that could smother living corals. Yet this daytime productivity comes with vulnerability at night. Unlike many reef fish that dart quickly into complex shelters, parrotfish often rely on specific sleeping sites and display limited mobility once settled. Their body shape, robust and laterally compressed, is not optimized for rapid nocturnal escape. In a reef environment where olfactory and lateral line cues guide predators through darkness, simple visual camouflage would offer limited protection. Evolution's solution lies in chemistry. The mucus cocoon is produced by specialized glands and extruded in sheets that expand as the fish rotates slowly within its resting cavity. The secretion forms a continuous envelope, open only at narrow apertures for water exchange. While transparent and nearly impossible to detect visually in low light, this barrier alters the diffusion of chemical signals. Fish constantly release metabolic byproducts and amino acids into the water, creating scent trails that predators can follow. By enclosing itself within mucus, the parrotfish reduces the outward flow of these cues, effectively muffling its chemical signature. The cocoon acts as a diffusion buffer, delaying and diluting scent dispersal into surrounding currents. In a habitat where sharks and eels rely on acute olfactory sensitivity to locate resting prey, this chemical masking can mean the difference between survival and predation. The mechanism extends beyond scent reduction. The cocoon may also serve as a mechanical barrier against ectoparasites, particularly nocturnal gnathiid isopods that seek out resting fish to feed on blood and tissue fluids. By interposing a layer of mucus, the parrotfish reduces the ability of these parasites to attach during vulnerable hours. In this sense, the cocoon represents a multifunctional adaptation, combining predator avoidance with parasite defense. The thin envelope becomes both shield and filter, a nocturnal armor tailored not for impact but for invisibility. Observing this process within the reef at night reveals a delicate choreography. The parrotfish selects a crevice aligned with prevailing currents, ensuring that water continues to pass gently across its gills even as it remains enclosed. Its respiration slows, metabolic rate decreasing as activity ceases. Within the cocoon, the fish remains motionless, suspended in a microenvironment where oxygen exchange continues, but turbulence is dampened. Outside, the reef vibrates with subtle movement. A moray eel threads its way through coral corridors, tongue flicking to sample chemical gradients. A reef shark glides above, scanning for the faintest disturbance in sand or water flow. The cocoon does not render the parrotfish invulnerable, but it reduces the probability of detection to a threshold that favors survival over repeated nights. Thermoregulation during these resting periods intersects with habitat choice. Reef temperatures decline slightly after sunset, particularly in shallow lagoons exposed to daytime solar heating. By retreating into coral recesses, parrotfish experience more stable thermal conditions, buffered by the reef's mass. The cocoon itself, though thin, may reduce convective heat loss by slowing water flow immediately adjacent to the skin. While not a primary function, this microclimatic effect contributes to maintaining metabolic equilibrium during inactivity. In tropical systems, where temperature fluctuations are modest yet biologically significant, even slight stabilization can influence energy conservation. Reproductive cycles and social dynamics add further context to this nightly ritual. Many parrotfish species exhibit sequential hermaphroditism, transitioning from female to male as part of complex social hierarchies. Dominant males often display more vivid coloration during the day, defending territories that encompass feeding grounds and resting sites. At night, however, these hierarchies dissolve into shared vulnerability. Both dominant and subordinate individuals create cocoons, their daytime distinctions irrelevant under cover of darkness. The shared strategy underscores a universal constraint imposed by nocturnal predators, one that transcends social rank. Migration patterns among parrotfish are generally localized, with individuals maintaining defined home ranges centered around productive reef patches. Seasonal shifts in water temperature or algal abundance may prompt modest relocation, but nightly movements remain consistent, returning to familiar crevices. This site fidelity suggests that suitable resting locations are limited and valuable. Coral architecture must provide sufficient enclosure to anchor the cocoon without exposing the fish to excessive current. Degradation of reef structure through bleaching or physical damage therefore has implications beyond feeding grounds, potentially reducing the availability of secure nocturnal refuges. Predator-prey interactions occasionally reveal the cocoon's limitations. Some moray eels have been observed biting through mucus envelopes when guided by other cues, such as minor body movement or residual scent leakage. In such cases, the cocoon may tear easily under force, offering little physical resistance. Yet even partial masking may reduce detection frequency enough to confer evolutionary advantage. The strategy operates on probability rather than perfection. In ecosystems where survival often hinges on marginal gains, a reduction in detection likelihood across successive nights translates into significant reproductive success. The biochemical composition of the mucus itself invites further scrutiny. Rich in glycoproteins, it forms a cohesive yet permeable matrix that adheres to the fish's body without suffocating it. The production of such material requires energetic investment, diverting resources that could otherwise support growth or reproduction. The persistence of this behavior across numerous parrotfish species indicates that the protective benefits outweigh these costs. It represents a calculated allocation of energy toward reducing predation risk during periods of inactivity. As midnight approaches, the reef settles into a quieter rhythm. Plankton drift upward in DL vertical migration, feeding under cover of darkness. The parrotfish remains encased, a silent occupant within coral stone. Its vivid scales, so striking under sunlight, now muted within a translucent veil. The cocoon catches faint reflections from bioluminescent flashes in the distance, yet it remains nearly invisible against the reef's textured surface. To an approaching predator, there is little to distinguish this recess from countless others. Chemical cues dissipate slowly, diluted by the envelope's barrier and the surrounding current's dilution. With the return of dawn, light filters gradually through the water. The parrotfish stirs within its enclosure, A quick flick of the body ruptures the mucus sheath, which disintegrates into wisps that drift away on the tide. Freed from its nocturnal shell, the fish emerges, resuming its role as a daytime grazer. The reef brightens, and the cycle repeats. What transpired in darkness leaves no trace beyond the ephemeral membrane now dispersed into the sea. The night disguise of the parrotfish illustrates an expansion of camouflage beyond visual mimicry in an environment where predators exploit multiple sensory modalities. Survival demands countermeasures that address more than appearance. By suppressing chemical signals and creating a microenvironment that dampens scent diffusion, the parrotfish navigates a world where invisibility must operate across sensory dimensions. The strategy reveals an ecosystem defined not only by color and form, but by currents of scent and subtle gradients imperceptible to human observers. Within the reef's nocturnal theater, concealment becomes an act of chemical restraint, a reminder that camouflage extends beyond sight into the invisible currents that shape marine life. The reef wall glows with improbable color. Fluorescent sponges encrust the limestone in saturated oranges and purples. Tunicates swell in translucent blues, and encrusting corals spread in neon greens that seem to hum beneath the filtered sun. Among this riot of pigment, a small form inches forward with deliberate calm. It is no larger than a human thumb, its back crowned with branching serrata, tipped in electric blue, its body banded in white and black, so sharply defined that it appears painted rather than grown. Nothing about it suggests concealment. It does not flatten against shadow or burrow into sand. It advances in full view across the reef face, a bright signal against an already extravagant background. Yet the longer one observes the scene, the more the outline dissolves into context. The reef itself is toxic in places, saturated with chemical defenses deployed by sponges and corals to deter predation. In this environment, brightness does not announce vulnerability. It announces risk. The sea slug's strategy is not disappearance, but alignment with danger. Sea slugs, particularly nudibranches, inhabit some of the most chemically complex ecosystems on Earth. Coral reefs are not passive structures, but arenas of biochemical warfare. Sessile organisms compete for space by releasing compounds that inhibit growth or deter grazers. Sponges produce toxins that discourage fish from feeding. Soft corals exude distasteful chemicals into surrounding water. Within this landscape of chemical arms races, certain sea slugs have evolved to exploit the defenses of their prey. Many species feed on sponges, hydroids, or cnidarians, ingesting toxic compounds and incorporating them into their own tissues. This process, known as sequestration, transforms the slug's body into a repository of borrowed defenses. The brilliant colors that cover their dorsum are not decorative flourishes, but signals, advertisements of unpalatability. A predatory wrasse cruising along the reef may detect movement in its peripheral vision. The sea slug's slow glide across coral would normally invite inspection. Fish are opportunistic, sampling unfamiliar shapes to assess edibility, yet experience shapes behavior. Fish that have bitten into toxic prey and suffered irritation or nausea quickly learn to associate specific color patterns with discomfort. The high contrast bands and vivid hues displayed by many nudibranches function as aposematic signals, warning predators of chemical retaliation. The slug's survival depends not only on its own toxicity, but on the memory and learning capacity of its potential predators. Camouflage here operates through psychology rather than concealment. The integration of borrowed toxins into slug tissues involves complex physiological pathways. As the slug consumes a sponge rich in defensive compounds, specialized cells isolate and store these chemicals without succumbing to their effects. Some species concentrate toxins in their serrata, the finger-like projections along their backs. When threatened, the slug may flex these structures outward, presenting the most chemically fortified regions to an attacker. In certain cases, nudibranchs feeding on cnidarians, such as hydroids, incorporate functional stinging cells, or nematocysts, into their own tissues. These kleptoconidae, stolen weapons, remain capable of discharge, providing an additional layer of defense. The transformation of prey weaponry into personal armor exemplifies evolutionary opportunism at its most precise. Coloration among sea slugs often mirrors the toxic organisms they inhabit. A species feeding exclusively on a bright orange sponge may adopt similar tones, blending visually into the substrate while simultaneously advertising toxicity. This dual function complicates the traditional dichotomy between camouflage and warning. In some contexts, the slug is cryptic against its specific host, merging into the patch of sponge it grazes. In others, when moving between patches, its brightness signals danger. The strategy is context-dependent, shifting along a spectrum between concealment and declaration. The reef's visual complexity amplifies this effect. In a habitat where intense pigmentation is commonplace, distinguishing a slug from its background requires close scrutiny. Mimicry introduces an additional dimension. Certain non-toxic sea slugs adopt color patterns that resemble genuinely toxic species, a phenomenon known as Batesian mimicry. By imitating the warning signals of chemically defended relatives, these species reduce predation risk without investing in toxin sequestration. However, this deception depends on predator learning remaining intact. If mimics become too abundant relative to truly toxic models, predators may begin to ignore the warning coloration, eroding the system's effectiveness. Thus, the balance between model and mimic shapes the evolutionary stability of these vivid displays. Camouflage, in this sense, becomes a dynamic negotiation between signal honesty and opportunistic imitation. The slug's locomotion reinforces its strategy. Movement occurs through muscular waves along a broad foot, producing a smooth, gliding advance. There is no sudden dart or frantic escape. This slow pace reflects both physiological constraint and confidence in chemical defense. Rapid movement might attract attention or disrupt the visual alignment with surrounding colors. Instead, the slug progresses deliberately, feeding as it moves. Its radula, a ribbon of microscopic teeth, scrapes sponge tissue with methodical efficiency. The act of grazing does not merely sustain the slug, but perpetuates its chemical arsenal. Each mouthful contributes to its toxic reserve, reinforcing the credibility of its warning display. Thermoregulation in these ectothermic invertebrates is mediated by habitat selection. Nudibranches often inhabit shaded reef walls or deeper zones where temperature fluctuations are moderated. However, some species tolerate shallow waters exposed to intense sunlight. In these settings, body coloration may influence heat absorption. Dark pigments can increase solar gain, potentially accelerating metabolic processes in cooler currents. Conversely, lighter patterns may reflect excess radiation in sunlit shallows. While color's primary function lies in signaling, its interaction with thermal dynamics underscores the multifaceted consequences of pigmentation. Reproductive behavior among sea slugs reveals further intricacy. Most nudibranches are simultaneous hermaphrodites, possessing both male and female reproductive organs. When two individuals encounter one another, they may exchange sperm in reciprocal mating, maximizing reproductive output in sparsely populated environments. Egg ribbons, often vividly colored and intricately coiled, are deposited on substrates near food sources. These egg masses sometimes contain toxins derived from the parent's diet, providing embryonic defense against predators. Even at the earliest life stages, chemical strategy shapes survival. Predator-prey interactions around sea slugs highlight the interplay of learning and risk. Juvenile fish lacking experience may attempt to sample a brightly colored slug, only to recoil at the chemical defense. Such encounters reinforce avoidance behavior in future interactions. However, certain specialized predators, including some species of flatworms and other nudibranches, have evolved tolerance to these toxins, targeting sea slugs despite their warnings. This selective pressure drives diversification of chemical compounds and color patterns, fueling an evolutionary arms race within a landscape already saturated with chemical competition. The reef at midday pulses with light, and the sea slug's colors intensify under direct illumination. What appears gaudy from a human perspective aligns seamlessly with the chromatic richness of sponges and corals. In shadowed crevices, the same patterns fragment under dappled light, making the slug's outline harder to resolve. The reef's complexity allows boldness without exposure. Against this backdrop, invisibility through brightness becomes plausible. The toxic illusion rests on the premise that warning signals and environmental context converge to confuse perception. As dusk approaches and colors flatten into subdued blues, the slug continues its measured progression. Its brightness remains, but contrast shifts. Predators reliant on vision retreat, replaced by those guided by scent. Yet the slug's chemical arsenal remains active, deterring sampling even in low light. In this transition between sensory worlds, the slug occupies a niche secured not by speed or stealth, but by chemical credibility. Camouflage is often imagined as blending into background until the eye cannot detect form. The sea slug challenges this assumption. In the saturated theater of the reef, survival sometimes demands the opposite approach, becoming so conspicuous that predators recognize and avoid. Deception here is layered. Some species genuinely toxic, others mimicking, all participating in a visual language shaped by chemistry. The toxic illusion transforms brightness into shield, spectacle into strategy. In the final light of day, as the reef hums with unseen chemical exchanges, the sea slug persists in open view, a vivid testament to the idea that concealment can emerge not from absence, but from a signal so strong it becomes untouchable.